Understanding the role epigenetics plays in cancer initiation and progression will be critical for future advances. Today, researchers need tools to understand how gene expression is altered in many types of cancer due to epigenetic changes. This video highlights the role of histone modifications and epigenetic regulation in the progression of breast cancer. Here are 4 targets related to epigenetics to consider for your breast cancer research. Phosphorylation of estrogen receptor alpha regulates transcriptional control of gene expression. Mutations in estrogen receptor alpha can confer resistance to treatment with tamoxifen. The protein EZH2 is overexpressed in many cancers, including breast cancer. EZH2 silences expression of genes required for the maintenance of cell identity by adding methyl groups to histone H3 lysine 27. UTX is a counterpart to EZH2 that removes methyl groups from histone H3 at lysine 27 and may enhance proliferation and migration of cancer cells in ER-positive breast cancer. Overexpression of EZH2 or mutation of UTX can change the amount of trimethylation at histone H3 lysine 27. Both hypermethylation and hypomethylation have been implicated in different breast cancers. Look for changes in methylation using trimethylhistone H3 lysine 27 antibody. Our portfolio of validated antibodies covers disease-relevant targets and enables your experiments in CHIP, IHC, and other applications. As you investigate epigenetic drivers involved in cancer, move your research forward with tools from cell signaling technology. Learn more